All right. So, this is a very broad overview of the next section. This is where the virus is actually going to enter and then do fusion. And they're actually two separate things. So, the virus is going to get pulled intact into the cell in a thing called a clathrin coated pit. And clathrin is just the name of a protein. And it makes a little cage around part of the membrane, and as it does, it binds to the membrane, and so it takes a flat membrane, and by forming a cage, it forms a little bubble, and then Dynamin is actually going to come up here at the top and pinch off the top of that, and so this is how the cell brings things inside. This is how the cell eats protein and stuff, and takes in some messages from the outside. And so when HIV interacted with CCR5, it set off this cascade. It's a signal that we caught one. It's just like a little, if you're fishing, you feel that little pull on the rod. It's like, ooh, time to reel it in. And so the cell reels it in, but instead of catching a fish, it catches an HIV. Yeah, which is terrible. And so the clathrin-coated vesicle is going to bring this in, and then once it's done its job, the clathrin actually disassembles, and the early vesicle is going to sort of merge with other vesicles. And there are all kinds of proteins that help do this, but that's more like a cell biology course. So this just happens the same way as it happens in a normal cell. Then, now that it's in the early endosome, that next compartment where the clathrin-coated pit merged with what are basically other clathrin-coated pits, the early endosome has a protein on it called the vacuolar ATPase. So that's what the V stands for. And this is going to use ATP to actually pump hydrogen ions, which is basically just protons, naked protons, into the lumen of the endosome. So it is going to acidify the inside of the endosome. And so this is called acidification of the vesicles. And now you may have seen a protein that looks like this before. Can you think where? Maybe when somebody was talking about mitochondria or ATP generation. Does that ring a bell? This protein looks like, and is actually very closely related to, the ATP synthase in your cells that you would find on the mitochondria. The top, consisting of the alpha and beta subunits, is going to actually physically spin around like a little spinny lollipop. The lowercase a here holds onto the sides. This is like the stator. This is like the rotor of their little motor if you're into engineering and stuff like that. And the bottom down here is going to have a slot in it, which is going to accept a little proton on one side in the cytoplasm and then deposit the proton into the lumen. So it's basically running backwards from the way that the ATP synthase runs. And since it runs backwards, it ends up using ATP instead of generating it, and so that's fine. It's an ATP using machine that will acidify the vesicles. Now, a good reason why you acidify vesicles is the same reason why you have acid down your stomach. It helps to digest food. So when a cell takes in proteins, and its own proteins, and wants to recycle them, a good first step is to acidify, because acidification will help break down the protein. And specifically, it'll do a particular thing to one amino acid that you find on just about every protein. It's called histidine, single letter amino acid code H. And so histidine can exist in multiple states, but one of those states is that at neutral pH, it'll just sort of spin here, and this sort of pentagon-shaped side chain will spin around. But then if there are a lot of protons, a lot of hydrogen positive ions around, they can actually interact with the distributed electrons that you would find running around this ring. And you can actually bind a positive charge to this, which means that now our molecule will have a positive charge. Over here, it's actually got a fairly hydrophobic side chain, and so it's going to go from hydrophobic to not just hydrophilic, but literally positively charged. And that means that this hydrophobic part was probably down, tucked away inside of a protein, next to other hydrophobic parts. And now that it becomes positively charged, it is going to be repelled, basically thrown out of the group. Just like when somebody in your group, picks up some new idea and starts spouting off, and yeah, boom, boot them out. You don't have to, but yeah, I've heard that it happens. And so this will basically end up turning the protein inside out. It'll pop it like a piece of popcorn. Yeah. Apologies to Orville Redenbacher. Okay, so this is what you've been waiting for. This is the mechanism. And so this is where all that learning that we've done up to this point is finally going to pay off. Oh my gosh. Such a long walk for such a great payoff. Yeah. Wow. Rarely do the stars align in this way. All right. So early on, you've got GP160. At some point, it gets cut. And the truth is, it can get cut by many different enzymes at many different points. They're all going to be furin-like enzymes, and none of the rest of this happens until that is cut. So it could happen theoretically on the cell that HIV is trying to enter, or it could happen at some stage before that, and it works fine either way. Don't worry about it. HIV is not worried about it, so you don't have to. That cut, of course, frees our dangly fusion peptide. So now it's just going to be out there wiggling and getting ready to do something bad. Yeah. Next step will be the attachment. So there's domain 1 of CD4 reaching around the side to poke on that little button, which is phenylalanine number 43. That causes the V3 loop to pop up, and the V3 loop will then be inserted into the co-receptor. This is our CCR5 protein here. And when that happens, it basically just locks GP120 onto the two receptors. So the whole point of binding all these things was to get GP41 into the right position to be able to unleash havoc on this cell. GP120's entire point is to get GP41 in the right place and then to get out of the way. Like, you can get there and then get off. Yeah, we do not need you. And so that's another reason why it's good that GP120 is cleaved off, so it can get all the way out of the way and not be in the way. Then you'll see that GP41 is going to unfold. And so it's going to reach up. That fusion peptide is going to be very happy if it can find a cell to spear. And GP120 got us close enough that, so conveniently, it's right there next to a cell. And so, bam, it springs up, and it's just like a knife going into that cell. And now the glycoprotein, the envelope protein, actually just GP41, is hooked at the bottom into the virus, and it's hooked at the top into the cell. Uh-oh, this is bad for the cell, right? Yeah, you bet it is. And so now we have basically stabbed up the cell. We've shivved it. The next step's going to happen. So this is going to involve heptad repeat 1 and 2, which is the yellow and blue on here. And remember, heptad repeat 1 and 2 are heptad repeats because they're not hydrophilic, not hydrophobic, but amphipathic. So they've got a side, which is actually out here on the outside of both of those things, that is real hydrophobic and is, I like to think of it as being real unhappy right now. And so what does it do? The two hydrophobic parts are going to come together. And again, we're going to bring these people that don't get on with their environment together, and they can talk to each other, just like on the internet. Oh, isn't it great? Yeah. All this stuff exists at the micro level and the macro level. And so heptad repeat 1 and 2 are going to come together, and they're going to form what is called the 6-helix bundle. Because remember, there's 3 copies of envelope that make a spike, and they have 3 heptad repeat 1s and 3 heptad repeat 2s. And they basically just fold down on each other and make a little bundle like that. There you go. And at this point, the MPER is now going to be active, and it's going to do that little trick of splashing some lipids up. That's our best understanding of what it does. Nobody's actually captured this in action. You can just show that it has to be there for this to happen, and this does happen when it's there, and it happens pretty quickly, and this is involved. Yeah. Eventually, we'll know more. Need more HIV research. It's going to be the only time you ever hear me say that. And so once we've splashed up enough of these lipids and pulled down enough of those lipids with the fusion peptide, we've now got to a state of hemifusion, which is where one leaflet, which is like one side of each lipid bilayer, is actually going to be fused together. So lipid bilayer on the cell and lipid bilayer on the virus, 2 layers of phospholipids with their heads pointing out and their tails pointing in. Hemifusion is where we merge those 2 layers. And so you've got still these 2 layers, they're intact. This is just the cells, this is just the viruses, but the middle layer is actually continuous between both. And that is halfway to fusion. And all the virus has to do is hold these in a hemifusion position. And then nature and the laws of physics will cause this to resolve into full fusion, because that is now the most energetically favorable thing. This is the case over and over with glycoproteins, with enzymes. The proteins just change the environment enough that it becomes difficult to do anything but the right thing. And if you think about it, maybe that's a metaphor for teaching. I don't know. You just put students in a difficult enough environment where the only real option, or the easiest option, is to do the thing that needs to happen anyway. So there you go. Maybe it's not just viruses that are terrible. Maybe everything's terrible. Maybe. All right, so that's enough for this section. We have one more little tiny section after this, I think. Yeah. And then we're done. But if we don't, and I miscounted those, then see ya.